So my name is Lei Jing. I'm working at National Institute for Physiological Sciences. I would like to thank Sophion for inviting me and organizing such wonderful symposium. Welcome to my talk titled Physiological Significance of the Interaction between TRPV3 and ANO1 or TMN79 in skin keratinocytes. So this is a two-part talk about TRPV3 ion channel. In the first part, we've reviewed that interaction between TRPV3 and ANO1 could positively regulate wound healing in keratinocytes, which comprise over 90% of the skin cells in the epidermal layer. I'll go through the background quickly and explain what we've done in detail. The TRP classes of ion channel consist of 6 subfamilies, which cover 27 TRP members. And each TRP channel consists of 6 transmembrane segments, and it is generally believed that 4 such structures together could form the homo or hetero tetramers. Almost all TRP channels are non-selective cation channels. TRPV3 is a warm sensor. In addition, it can be activated by some compounds derived from the nature plants as camphor and some more synthetic chemical as 2-EPB. Particularly, TRPV3 is highly expressed in the skin keratinocyte and hair follicles, which prompted a lot of study focused on how the dysfunction of TRPV3 could contribute to the skin homeostasis and regulate the skin, cause the skin diseases. For example, TRPV3 is required for the formation of skin barrier, wound healing, hair growth, itch, as well as the temperature sensation, although this contribution has been controversial. TRPV3 is also clinically related to some skin diseases, including Olmsted syndrome, which is mainly caused by the single glycine mutation, and atopic dermatitis, psoriasis, and pruritus. Including TRPV3, many TRP channels, especially the TRPV family, are highly calcium permeable. Therefore, this calcium influx upon the channel opening could lead to the activation of the calcium-sensitive intracellular processes or some calcium-dependent ion channel activities. The ANO1 is a calcium-activated chloride channel, which controls multiple functions, including the neuron excitation, muscle contraction, cell migration, and volume regulation. So we've previously reviewed an interaction model between ANO1 and several TRP channels. For example, TRPV4 together with ANO1 could regulate the separation of the cerebrospinal fluid, saliva, and teeth through the water channel. In addition, TRPV1 and TRPA1 could sensitize the ANO1, which further potentiates the nociceptive signal through the chloride efflux. Therefore, we make hypothesis that this functional interaction could work in many tissues in which TRP channels and ANO1 are co-expressed. We decided to focus on TRPV3. As we normally do for ion channel, we overexpressed TRPV3 and ANO1 in HEK293 cells and perform whole cell patch clamp with the NMDG chloride solution. So this solution is usually used to prevent the current from the rich cations. By applying a TRPV3 agonist, the chloride current was clearly observed in the cell expressing both TRPV3 and ANO1, but not in those expressing either of them. So this suggests the calcium influx through the TRPV3 could activate ANO1 and cause the chloride fluxes. In addition, we performed a co-IP showing that these two proteins likely make a complex. Given the enrichment of TRPV3 and ANO1 in the skin keratinocyte, we successfully observed the camphor-induced chloride current in the normal human epidermal keratinocyte cell line, NHEK cell. And this reversal potential was shifted to a positive value when we replace the extracellular chloride to 4 millimole. In addition, the ANO1 specific inhibitor, ANO1, inhibited the camphor-induced chloride current. So this suggests the interaction between TRPV3 and ANO1 in the human keratinocyte. More than that, we also confirmed the interaction between two proteins in the primary mouse skin keratinocyte, as shown the camphor-induced chloride current in the wild-type keratinocyte, but not in those lacking TRPV3. Likewise, ANO1 inhibited the camphor-induced chloride. So we next ask, are there any physiological significance based on their interaction? Both proteins were previously suggested to be important for the cell proliferation and migration in different tissue cells. Therefore, we performed Wood-Healing assay to examine their interaction in the skin keratinocyte. And the images were captured at 12 and 24 hours after peeling off these two chambers. We first examined the contribution of TRPV3 by using its inhibitor dicloning, and the cell migration was inhibited by the 50 micromole dicloning at 12 hours and by the 100 micromole dicloning at both 12 and 24 hours, suggesting the involvement of TRPV3 in the cell proliferation and migration in the human keratinocytes. The cell migration was also suppressed by the ANO1 inhibitor T16A at both 12 and 24 hours, suggesting the involvement of ANO1. So the contribution of ANO1 likely implies the significance of the chloride ion. Therefore, we performed the assay in the low chloride containing medium. And similar as the inhibition of TRPV3 and ANO1, the low chloride medium also inhibited the cell migration at both 12 and 24 hours, suggesting the involvement of chloride in the cell proliferation and migration. Although I did not mention many details, but we concluded here the calcium through the TRPV3 could cause the activation of ANO1 and lead to the chloride influx. So this further regulates the cell proliferation and migration by keratinocyte. So we believe this TRPV3 and ANO1 interaction could be an important molecular mechanism for wound healing in the skin. Let's move on to the second part. We identified another interaction model between TRPV3 and membrane protein TMEM79 in skin keratinocytes. It is generally believed that the thermal information from the skin is conveyed through the sensory nerve ending embedded in the skin epidermal layer. Moreover, the skin keratinocytes are also important to detect the ambient temperature. Our previous study reported that TRPV3 in keratinocyte transmit the temperature information to sensory neuron via releasing ATP. However, the in vivo role of TRPV3 in the thermal sensation is still controversial. TMEM79 is a less studied membrane protein with 5 transmembrane domains. Similar to TRPV3, it also had expressed in the skin keratinocyte and hair follicles and contribute to the epidermal integrity and skin barrier function. Interestingly, the lack of TMEM79 could cause the similar skin illnesses as the gain-of-function mutation of TRPV3, including the dermatitis, hair loss, and itch. Considering such functional interaction and functional similarity between TRPV3 and the TMEM79, we examine the possibility for their interaction. Again, we overexpressed TRPV3 alone and both TRPV3 and TMEM79 and performed the patch clamp in HEK293 cell. We found the TMEM79 reduced the 2-EPB induced TRPV3 current, as shown here. And this negative regulation is in a dose-dependent manner with the identical EC50. Therefore, these changes in efficacy but not potency suggested that the difference in current amplitude is caused by the difference in the expression level of TRPV3. However, TMEM79 did not affect the GSK101 induced TRPV4 current. So TRPV4 is another keratinocyte abundant TRP channel. So this suggests the specificity of the TMEM79 to TRPV3. To answer how the TMEM79 could regulate the function of TRPV3, we performed the biotemylation IC in the overexpression system and found the TMEM79 could reduce the TRPV3 current in both whole cell lysate and the plasma membrane level. So together with the previous patch clamp result, we conclude here the TMEM79 decreased the TRPV3 mediated current through reducing the expression level of TRPV3 on the plasma membrane. And in line with that, we also found the TMEM79 changed the translocation, the TRPV3 from the plasma membrane to the cytosolic side. So this is consistent with our previous result. And more than that, in many cells expressing both TRPV3 and the TMEM79, we observe the co-localization of the TRPV3 and a lysosome marker here. So this likely implies the TMEM79 could cause the TRPV3 degradation. So there are two major ways for the protein degradation pathway. One is a ubiquitin proteasome system. Another one is a lysosome protein lysis pathway. So in light of that, we incubate the cell expressing either TRPV3 alone and expressing TRPV3 and TMEM79 with two lysosome inhibitor and two proteasome inhibitor here, and found only two lysosome inhibitor could rescue the protein reduction caused by TMEM79, although the two proteasome inhibitor did not. So this indicates that TMEM79 promotes TRPV3 degradation in the lysosomes. So there's another possibility that the reduced TRPV3 expression likely caused by the lyse protein synthesis. So here we use a protein synthesis inhibitor, cycloheximide, and incubate in the cell at different time points. And we found the TRPV3 degrade much quicker, especially at 24 hours when co-expressed with TMEM79. This really suggests the lyse TRPV3 expression caused by TMEM79 is due to the more protein degradation rather than the lyse protein synthesis. The functional interaction between TRPV3 and the TMEM79 suggested a possible physical interaction. By performing the co-IP, we confirmed their physical interaction as shown here. In addition, we performed the proximity ligation assay. So the localization of the magenta puncture here reflects the protein interaction likely located on or near the plasma membrane. So what about the interaction in the skin keratinocytes? Here we isolated the keratinocyte from the mast-cell with high purity and performed the whole cell patch clump by applying a TRPV3 agonist and the TRPV4 agonist. And we found there are over 80% of the keratinocytes could respond to both TRPV3 and the TRPV3 agonist and the TRPV4 agonist. So this suggests most of the small skin keratinocytes functionally express both TRPV3 and TRPV4. By comparing the current amplitude induced from the 2-EPB and the GSK101 in wild type and the TMS79 knockout keratinocyte, we observed the 2-EPB induced a significant larger current in TMS79 knockout mice compared with the wild type. But GSK101 induced current is not largely different between them. So this is in line with the result we acquired in the HEK293 cells, in which the overexpression of TMS79 downregulates the function of TRPV3. And the keratinocyte liking TRPV3 here used for checking the specificity of the 2-EPB. As I mentioned in the early slide, it is still controversial whether the TRPV3 is involved in the warm sensation. Here we use a thermal gradient ring with duplicate thermal gradient from the 10 to 45 degree. And this thermal gradient ring provides more advantages as compared with the linear one used in many other studies, and give us the more accurate preference information and can eliminate the effect of the corner. So by comparing the spending time, we could see compared with the wild-type mice with the strongest preference around 32 degrees, the TRPV3 knockout prefer a lower temperature and shows the less difference at around the warm temperature range, but identical for the noxious cold and noxious heat compared with the wild type. So this really indicates TRPV3 does involved in the warm sensation. Interestingly, the TMS79 knockout mice spent nearly half of their time at a higher temperature, about 35 degrees. So we assume this behavioral preference is caused by the increased TRPV3 activity in the TMS79 knockout keratinocytes. Here is a summary. In this study, we found that TMS79 could downregulate the function of TRPV3 and cause the TRPV3 degradation in the lysosome. And this interaction between TRPV3 and TMS79 could be involved in the regulation of the warm sensation. At last, I would like to thank for Professor Makoto Tominaga and all the lab members from the thermal biology group and our collaborators, especially for Dr. Yamanoi for his main contribution in the first work. So I'd like to take the question next.
Okay, thank you very much.
Thank you for beautiful presentation. And you showed V3 is the surface plasma membrane expression of V3 is decreased and accumulated in lysosome when TMEM79 is co-expressed.
Yes.
And what's the mechanism? Why does it happen?
So far, because we have no any related experiment like related to your question, but we make the I can make the hypothesis that because the TMS79 is an ER resident protein, so it's likely cause the folding, the misfolding of the TRPV3 protein and lead to the lysosome for further degradation. But this is just my hypothesis. We have no any.
Does TMEM take V3 to lysosome?
We actually did not check whether TMEM79 localized with lysosome.
I see.
Yeah, but it's mainly located in the ER.
In your PLA data, the signal was observed only on the plasma membrane, but not in internal organs. So, and I thought it could also accumulate together in organelles, but that's not the case?
Yeah. If you see this result, it's actually mainly located on the membrane or close to the plasma membrane, but there's more portion located inside. So I'm thinking this might be some intermediate process, like the TME79 drives the TRPV3 from the plasma membrane and move to the inward side and probably go to ER first and then lysosome and cause a degradation. So this is just my hypothesis. Okay, thank you.
Yeah, thank you very much, Kim from Korea. It's really interesting. And first part of your talk, the interaction between ANO1 and V3, you intentionally use the BAPTA as a pipette in the pipette solution to limit the calcium diffusion distance, I think. So you showed that functional interaction, very close interaction. Have you ever so this is the molecular proximity proof of the experimental evidence. Is this slide?
Sorry, proof what?
This is the core IP data.
Yeah.
So but in the physiological conditions, we actually don't need such a strong calcium buffer. There are many calcium flux pathways that could finally affect ANO1. So why do you think is the V3 and ANO1 has a more specific relation? Or my question might be wrong. There could not be, so why? Is there any other calcium sources are also interacting with ANO1 function?
Yeah, as I mentioned in the introduction, ANO1 is a calcium activated chloride channel, and actually it can be direct activated with very low concentration of calcium. So the reason we use here just want to control the concentration of the calcium, like use a specific 100 nanomolar free calcium, and to avoid the other calcium resources.
Okay, thank you.
One possible.
Here. Jim, beautiful work. Here. So do you think that anoctamin directly binds to TRPV3? And if yes, have you tried to map the interface of interaction?
Sorry, you mean the TRPV3 bind to?
Anoctamin and ANO1.
Oh yeah, it means they bind.
Do they interact directly? Yeah.
Oh, sorry, we did not check their specific interaction in the cell, so I cannot make conclusion for this question.
Okay.
Yeah, but yeah, probably we can perform the same experiment I performed in the second part. Yeah.
Yep. Thank you.
Yes, possible.
Nice work.
Yes, and the last question up there, yes. No, sorry. Anybody else? Yes, here.
So my name is Hiroko Takechi from Osaka University. I have a question. So in native keratinocyte cell, so do you know how localization or channel density, expression density about the TRPV1 and ANO1 and TME79?
Okay. So I never checked the expression of anoctamin-1 in the primary skin keratinocyte, but based on but based on This is a weak point for our study. So there's no good antibody to for both TRPV3 and TMS17. So we could not detect their relative expression level. So how to say? The TRPV3, actually one previous study shows the TRPV3 likely make a gradient in the skin epidermal layer. So because the experiment the keratinocyte we use here is mainly basal layer of the keratinocyte. So we are not able to check the relative TRPV3 expression between each layer. So,
Okay.
yeah.
So it is difficult to isolate the skin, the keratinocytes. So I think, so if you use the isolated skin keratinocyte and excise the patch, and then you can see the single channel current and calculate the number of the channel and density.
Okay. Yeah, it's good suggestion. I agree. It's not so difficult to isolate the keratinocytes, so it's deserved to identify in the future.
Thank you very much.
Okay, thank you very much. And yes.